Have you ever wondered how all the food that we eat gets digested in our body? There are little molecules that do a lot of work that are called as enzymes. Enzymes are proteins that act as biocatalysts that help speed up chemical reactions like digestion and metabolism. They build some substances and break others down. All living things have enzymes. There are about 1,000 different enzymes in our body that work to keep us healthy and active. Enzymes are very specific and only react with chemicals called as a substrate. Today we shall be studying about an enzyme called as catalase. Catalase is a common enzyme found in nearly all living organisms exposed to oxygen. It is a crucial antioxidant enzyme in living cells, which plays an important role by breaking down hydrogen peroxide and maintaining the cellular redox homeostasis by protecting them from oxidative damage. Catalase helps in preventing the damage by breaking up hydrogen peroxide into harmless water and oxygen. This enzyme is present in commercial yeast. Today's experiment shall explain how this enzyme activity takes place and what makes the activity possible. The materials that we will be requiring for today's experiment are liquid dish soap, a tablespoon of dry yeast, hydrogen peroxide at 6% concentration, glasses, teaspoon, tablespoon, warm water around half cup, label makers, pen, tissue roll and some food coloring. We begin our experiment by preparing our yeast solution. Take a bowl of warm water and dissolve the dry yeast. Let the dissolved yeast rest for at least 5 minutes. Label the glasses from 1 to 3 and mention the amount of hydrogen peroxide that has been added. We add no amount of hydrogen peroxide to the first glass. We add half tablespoon and 2 tablespoons of hydrogen peroxide to the second and third glass respectively. This shall help us study the enzyme activity well. To all the labeled glasses, add 1 teaspoon of liquid dish soap. To each of the labeled glasses, add a drop of food color and stir well. This just makes the experiment more fun. After adding the soap and the color, mix this solution very well. As we will be handling hydrogen peroxide at 6% concentration now, be careful while using it. Any spill on your skin should be rinsed with plenty of water. As mentioned earlier, as per the label makers, we shall be adding the same amount of hydrogen peroxide to each of the glasses respectively. Nothing shall be added to glass 1. After adding the hydrogen peroxide to each of the glasses, stir well. As we come to the main segment of our experiment now, please place tissues under all of your glasses as this could get little messy and this will also make cleaning the entire setup easier. Add 1 tablespoon of the previously prepared yeast solution to each of the glasses and just observe the reactions. Are you wondering how this happened? The answer is, when the enzyme catalase comes in contact with its substrate, hydrogen peroxide, it starts breaking it down into water and oxygen. Oxygen is a gas and will always try to escape the liquid. The dish soap here that we added was able to trap the gas bubbles, which results in the formation of this stable foam. As long as there is enzyme and hydrogen peroxide present in the solution, the reaction continues and the foam is produced. If there is no hydrogen peroxide present, the catalase cannot function, which is why glass 1 had no bubble or foam production. The presence of hydrogen peroxide makes the catalase to be active as observed in the other glasses. In fact, the catalase reaction is dependent on the substrate concentration. If you have an excess of enzyme but not enough substrate, the reaction will be limited by the substrate availability. As a result, glass 3 had more substrate, so we observed more foam production. catalyst does not take part in the reaction itself. So, how does it really work? Each chemical reaction needs a minimum amount of energy to make it happen. This energy is called as the activation energy. The lower the activation energy, the faster it takes place. Enzymes have the ability to lower the activation energy of a chemical reaction. The compounds or molecules the enzyme reacts with are called as substrate. The enzyme pocket has a special shape so that only one specific substrate is able to bind to it. Once the molecule is bound to the enzyme, the chemical reaction takes place. These enzymes are very specific. On a cellular level, if there is imbalance of antioxidants, then the physiological condition is called as oxidative stress. A catalase is one of the crucial antioxidant enzymes that mitigates oxidative stress to a considerable extent by destroying cellular hydrogen peroxide to produce water and oxygen. The deficiency or malfunction of catalase can result in many illnesses such as diabetes, hypertension, anemia, vitiligo, Alzheimer's, Parkinson's, bipolar, schizophrenia, and cancer. I hope this video was able to help you understand about enzyme activity, the enzyme catalase, and its importance. Thank you for watching.